It's great to be here and hearing all these talks and actually be part of this this movement, I think, in terms of trying to help all of us do something about improving reproducibility of research. I think for Abcam, for us in Abcam is integral to the way we do we work and why we work in this area. If you ask me why I'm doing this and why in Abcam we are doing this, is because our mission is to help scientists and researchers in life sciences discover more faster. And we believe that providing them with reagents that work as they should work first time is basically the way to do that. At the same time, really, when we think about a lot of the reagents that we are developing now more and more are being translated either into the clinic or diagnostics. And then our responsibility is even greater because now these reagents could actually help save a life or actually potentially have the opposite effect if they are not specific. So I think it is really more than just helping reproducibility of research. It really, we should feel the responsibility that is even further than that. And today I want to talk to you about what we are doing, what we have been doing since 2 years ago when I presented here last time in terms of improving, working towards antibody reproducibility and really reagent reproducibility. And so we believe that this is a multifactorial effort. We cannot just concentrate in one area. And so the three main factors that I will focus today is I will discuss how what we are doing to improve and to confirm the specificity of our antibodies. Also, how we make our antibodies, the development and the manufacturing of the antibodies has a huge role to play here because it's not just how you validate them, it's actually how they are made. And also the support systems, the quality systems and capabilities and the transparency, because for us transparency is key to let people know when something is not working well and to also accept the feedback and put it there completely openly in our website when something is good or not good in terms of our products. So in terms of specificity, as I mentioned, this is critical to get results, accurate results first time. And we have a tailored approach for our antibody validation. And we decide what application we should test our antibodies depending on the area of research and when most scientists are going to be using them. As well as a lot of people have already described, for us, it's essential to make sure that the antibody works in the endogenous sample where it should be, where the protein is expressed naturally. In this case, for example, and for a lot of our histone antibodies, we test them on Western blot immunohistochemistry, immunocytochemistry, in peptide array, chip, and actually this is one area where we are now doing a lot more, and we are going to be we are working on developing a multiplex peptide array to actually do a lot more peptides in one single array. But this is probably for next time. I'll give you more data on that. Yes, I think for us we have been in this business for 20 years, and this is not new to us. We have been taking the effort to validate the antibodies that we have and go back and revalidate if we think that something is, it didn't have as much validation as we thought, or when we get feedback from researchers, we always go and corroborate that the validation has been done correctly. So I couldn't give this presentation without having a slide on the pillars, and we have been doing this one or many of the pillars for our antibodies from pretty much the beginning when we have developed antibodies in our lab. For me, there are some of these types of validations that are more important than others. I'm not a real fan of the independent antibody validation, and I'll show you why, because when we have been doing knockout validation and we do multiple antibodies to the same target, we are finding that 2 or 3 antibodies could be really horrible and actually are pretty bad. So if you selected 2 of those antibodies that have the same specificity or lack of specificity, that you really couldn't corroborate that they are both good. But anyway, I think doing in conjunction these multiple applications is actually a good thing. So, and as I was saying, we have been doing a lot of these applications, but about 5 years ago, we started really, sorry, not 5 years ago, about 3 years ago, we started doing the knockout validation at scale. I mean, we had been doing knockout validation whenever we could get access to the samples or knockout animals or collaborations, but we felt like this wasn't really enough, that we needed to do this at larger scale. And since 2015, we have been doing this. And we started really doing the validation in terms of mostly using one particular knockout cell line and doing Western blot or ICC. And now I will show you what we have been doing. This is what I showed you last year, pretty much. And now I will start describing what we have been doing since then. We have been confirming the antibody specificity in multiple applications, in the knockout, but in applications where we feel like people will be using these antibodies, as well as we have now started to implement this knockout validation on all our antibodies that are going to be part of assays, the simple step ELISAs or the multiplexing assays as well as very importantly, we feel like we need to do this in our pipeline. And so we started implementing the knockout validation within the discovery pipeline to actually help us fail faster. I think it is really important that we don't get to the very end of the development and find that the antibody is not specific. So this has helped a lot. So starting with the confirmation of the antibody specificity for the target, as I said, we have been doing multiple applications, and the way we do this analysis is to have 3, 4, 5 antibodies to the same target, and that helps us basically corroborate that the clone, knockout clone, is correct, and as well as eliminate, discard the possibility that some of those antibodies could be non-specific. And so we are doing now flow cytometry, ICC, as well as standard Western blot. And when one of these antibodies doesn't work, we move it out of the catalog, and we contact the customers that have purchased that antibody for the past few years. And we make a big effort to try to contact everyone so that they know that if they had published with this antibody, they know that it probably wasn't a good thing. It wasn't following the right target, I should say. So we have also tested antibodies that are top-sided, and we were slightly nervous, as you can imagine, testing our top-sided antibodies. And it's been great that a lot of these antibodies are very specific. And we are now we have now tested over 1,500 antibodies in this system, and because we are also testing benchmark antibodies and top-sided antibodies that may not be ours. Unfortunately, we actually have found that some antibodies that are top-sided are not really that specific. And this is a concern because most researchers purchase antibodies that are top-sided. And then when we need to basically realize that the top-sided antibodies may not be actually the best. It might be the oldest one, and that's why it has lots of publications, but it may not be necessarily the best. And this obviously happens a lot more with polyclonals than with monoclonals. Now moving on to antibodies being tested when they are part of an assay. It is as important for us, or even more important, that these antibodies that are going to be part of assays are actually validated and that are fully specific as well. And so these are just a few examples of the antibodies that we have tested that are part of simple step ELISAs and our multiplex vireplex ELISA assays. And these are three examples of antibodies that we have developed using the knockout technology in the pipeline. So when we make our rabbit monoclonals, we put this in the screening process to screen all of the clones on knockout. And then we actually realize early on that if the clones are going to be successful or not. And this is something that we want to do more and more. And we find that really could help us a lot from the very beginning identify if we should continue with that program or not. And as I was saying, a lot of the antibodies that we are developing are becoming part of a therapeutic research and discovery program, or are part of a diagnostic, companion diagnostics, for example. And to me, I find that this is a really important thing when this is actually something that is quite close to me, because my sister-in-law had cancer and was being evaluated to see if she would actually be able to respond to a PD-L1 therapy. And I found that basically if you have a non-specific antibody, you could actually be dictating if somebody is or not able to be part of a trial. It goes as far as that you basically need to realize that this is a big responsibility that we have. It's not just saying that this company is doing the right thing. So I think it's part of why we are doing this. And that's why I think it is so important that we are here trying to solve this situation. So I've told you what we have done so far with the knockouts. And now what we are doing in the next, we are up to 1,500. What are we going to be doing to get done next? 14,000 antibodies. And so we have been working with one particular cell line, CRISPR knockout cell line, and now we are expanding our collaborations, and we just signed a partnership with a partner, and we are moving to many different knockout models as well, and also to address what is being already discussed: essential proteins, multi-species. Obviously, there is conditional knockouts or temporary knockouts that we can be using in some instances. And we are now moving into really fully embed these knockout validations in the new antibody discovery pipelines. With that, I'm going to move to, wow, consistency now move to, it actually looks quite good. Obviously, consistency is extremely important to produce best-in-class reagents, and this is where the development is extremely important. How are we going to be developing these antibodies? And it is as basically proteins are an essential part of delivering high-quality reagents, and we are now increasingly making more and more of our proteins ourselves, and using them for our own antibody development. And we have now a group headed by Bert, who's here, and is really, we are putting as much effort on the quality of the proteins as we are making on the quality of the antibodies, because rubbish in, rubbish out, obviously. And so a lot of this is being done in a way that we want to have sustainable processes, and we have put a lot of high throughput equipment and automation in our manufacturing and our new product development pipelines, and this is something that I think we need to continue doing more, because we are seeing that that is actually helping us with a sustainable process. And another thing that is hugely important is the batch-to-batch QC. And for us, this is one of the reasons why recombinant antibodies are so important. And in the past few years, we actually decided to stop making any polyclonals, new development of polyclonals, because we believe that's not the future. We believe that the recombinant antibodies will be the future, and we decided to just concentrate our efforts on the recombinants. And we have now over 12,000 recombinant antibodies, and we develop them either from scratch using hybridoma technology and then converting them into recombinants, or through Axiomics technology, phage display. And this is a synthetic in vitro approach. So also, as well as developing our own antibodies, we have been licensing antibodies, and recently we licensed a portfolio from Roche of their Spring Bioscience antibodies, and which are really very good antibodies, which actually were developed with the RapMap technology as well. And this is to contribute to our effort to have the best reagents. And we put a lot of effort also on the batch-to-batch consistency, and we find that obviously the batch-to-batch consistency is much higher with recombinant antibodies than with a polyclonal, for example. And not that we don't like the polyclonals. I actually don't like polyclonals. I don't like. But they are so difficult to keep in stock and keep remaking them. Every time that you run out of a batch of polyclonal, you basically have to start from scratch, and it's making a new one almost, even if it is the same immunogen. So they are a pain, and I'm very glad that we are not making them anymore. So we are using a lot of these antibodies for our immunoassays, and this is another reason why our immunoassays are so robust. We find that we do actually a lot of tests as well in our immunoassays, not just our antibodies, that we dedicate a lot of resources. We have an entire team in Eugene, Oregon, that is dedicated just to develop immunoassays. And so obviously, I'm not going to go into details on how much effort we put on the validation of these assays, but it's part of our core. I don't know why all of these slides were messed up, but anyway. The last part really is to talk about the quality systems, our capabilities, and our transparency. And so we have a quality infrastructure that we have set up, and this is kind of to have that the basics of how we do things in Abcam. So we have a quality management system, ISO 9001 and ISO 13485 as well for our IBD programs. And we have a very robust process and rigorous testing, and we have a process to also get rid of products when they don't work. And that goes also into transparency and scientific support, which is actually, as I mentioned, if we are going to eliminate products from the catalog, we don't just list them from the catalog, we actually go and contact people. And the scientific support there is also to receive feedback and to send it to the lab for us to act on it. And the last part, and a really hugely important part of our success, is our people. And we have been hiring great people to work in Abcam to make this happen. And we have sites working on reagent and immunoassay development and manufacturing in 6 sites. We have around 400 people, and we are hiring a lot, actually. So if anybody's interested, we have a lot of open positions on all sites. Please go to our website. So I have already discussed with you the specificity, the consistency, and our systems in terms of quality. And the last part I wanted to discuss with you is just to leave you with an example of one of our antibodies that has been put into companion diagnostics. ARB7 is an antigen receptor splice variant, and Epic Sciences has developed an assay for the detection of to predict if a person is going to be responsive to a prostate cancer treatment or not. And again, I think this is, again, to leave you with the idea that this is our responsibility as much as reproducibility.
Oncotype DX, ARB7 nucleus detect, measures the most compelling biomarker in castration-resistant prostate cancer. What we know is that men who have cancers that express ARB7 no longer benefit from the hormonal therapies, but they still benefit from chemotherapy. So once you know a man has tumor cells that are ARB7 positive, you know that the right choice of therapy for those men that can still improve their quality of life and the duration of their life is chemotherapy.
Well, thank you very much. I'm happy to take questions.
Questions for Alejandra? Everything is crystal clear? No? Yeah.
So I was very interested in your moving strategy to not use chromosomes antibody. And at the moment you're showing that you will use them in flex application, if I understood. But are you planning to also validate them for other applications, let's say IHC, immunochrome?
Our recombinant antibodies? Yeah. Oh, we are actually, sorry, I didn't specify that. All of those antibodies have been validated in multiple applications, IHC, ICC, flow, whichever application it is closer to the area of research where people will be using them. The multiplexing is just, that is a different type of application that we are doing.
So do you use the same strategy as in knockout validation with all your clones that we get from the phage campaign?
Yes, we have implemented the knockout validation on the phage discovery as well, but the phage discovery is only a small proportion of our development at the moment. The largest proportion is the RAP map from hybridoma to recombinant. And again, yes, we are trying to put this in as many programs as we can, but the limitation is the availability of the knockout cell line.
Okay, first you.
Yes, do you have the recombinant antibodies all in one format, or can you do isotype switching easily, or do you have multiple constellations?
Yeah, we are working on that. So we can definitely do isotype switching, yeah. Not necessarily isotype switching because these are rabbit monoclonals, but we can put them in different backbones.
Necessary to change
The backbone, the framework, yeah.
Matt?
Do you ever see a situation where a knockout doesn't work, but you know the antibody's good?
How do you know the antibody is good?
Under experimental evidence?
We have talked about that.
Yes, I think when that happens, you have to really trust that the other evidence is stronger than the knockout, or to make sure that your knockout is good. So, and that's why we test multiple antibodies with the same knockout sample. But it is quite difficult, and we have had some customers that tell us, no, this antibody is great, and we are like, it's failing. So then we have to really go into, actually we had a really good example where the knockout cell line was knocked out on an area of the protein that didn't have the epitope. And so you have to analyze all those cases carefully and know if the knockout is actually going to be useful for that particular antibody.
Yes.
So can I invite you to tell us as much as you're able about the knockout validation 1500 set, where you had some failures? What scale of failure did you experience in that program?
So the scale of the failure was actually surprisingly low, based on the concept that 50% of the antibodies out there are rubbish. In our hands is not as high as that locally, and our rabbit monoclonals have the lowest failure rate. Obviously, polyclonals have the highest, but that you sometimes don't even need a knockout to know that they are not good. So, yeah.
So if you were looking across the industry, what scale would you think? Is it 10%?
No, I think
1%?
it really, because of polyclonals are so prevalent, I think, I'm not sure if I could say that it's closer to 50%, but I would say at 30%.
So I have a question for you. Now that you are expanding your knockout to, first you started doing a lot of Westerns, and now you're expanding it to other applications. How often do you find dissociation between the two? So that, let's say that you find that that you have a single band that disappears in a Western one, and then when you do a knockout or testing on immunofluorescence, you see that it's cross-reactive.
Very rarely.
Very rarely.
Yeah, we have had some cases where Western blot looked okay, but the immunocytochemistry didn't look good because it didn't seem to, well, probably was, I don't know, should have been not nuclear, and we were looking at cytoplasmic cross-activity or something like that. So when we have found those cases is in some instances where conjugation, with conjugated antibodies, for example, that we believe that maybe the conjugation has altered something in the binding, but it's not very common, which is good.
That's reassuring. Yes. You first.
We use a lot of the phosphorylated antibodies, so the antibodies target the phosphorylated proteins. Do you knock out, validate them for
Yes, yes, that's what we do. And again, with those, you need to have self-treatments of the knock. We actually do treat the knockout cell line with, I don't know, the particular treatment that will be inducing the phosphorylation, and then we see with that treatment we can evaluate that. Or we knock out the actual target, and then we do other assays that will tell us that if the specificity for the phosphorylation is there.
Like the phosphatase.
That's right. Mm-hmm.